Today, we're cracking a case of viral evolution. A team of scientists in India stumbled upon a flu virus that it had leveled up. It became super efficient at its job. But how? Let's follow the clues they uncovered.
It's a question that's absolutely central to public health, right? Viruses are always changing. They're constantly adapting, and sometimes they get way better at what they do. And this isn't just random luck. It's a process of tiny, tiny upgrades that can lead to some really big advantages. So how exactly do these upgrades work on a molecular level? Well, that's what we're here to find out.
So, our story kicks off with a real puzzle. Researchers in India were doing routine surveillance on the H9N2 bird flu virus, and they noticed something odd. One of their viral samples, one they isolated back in 2017, just wasn't acting like the others. It was an outlier, a total curiosity that basically screamed for a closer look.
And here it is, the very first clue. In a standard lab test, a flu virus latches onto red blood cells. Then, over time, it uses this little tool to cut itself free. But this one particular strain, it was a master escape artist. It was cutting itself free from the cells way faster than its relatives. This rapid escape, it's called elution, by the way, suggested it had developed a much more efficient tool for the job.
Okay, so with a mystery on their hands, the scientists basically turned into detectives. And what's their first move? Interrogate the genetic code, of course. They knew this difference in behavior had to come from a physical difference in the virus's makeup. It was time to find the suspects responsible for this high-speed getaway.
So let's meet our two key players. On the left, you've got H9N2 99321. This is a strain from 2009 that's kind of our baseline, the original slower virus. And on the right, H9N2 1726265, the 2017 strain that's our speedy mutant. The big question is, what makes this one so different? The investigation pretty quickly zeroed in on a key part of the virus, a protein called neuraminidase, or Na for short. The easiest way to think of Na is as the virus's escape tool. See, after a virus makes copies of itself inside a host cell, all those new virus particles have to break out to infect more cells. The Na protein acts like a tiny pair of molecular scissors, snipping the connection that holds the virus to the cell surface and setting it free. So it makes sense, right? A more efficient pair of scissors means a faster escape. And bingo! When the scientists compared the genetic blueprints for the Na protein in our two viruses, they found their prime suspects. The faster virus had this unique combination of 5 amino acid substitutions, 5 tiny little changes to the building blocks of its molecular scissors. And get this, these changes were located right near the protein's active site, the business end of the tool. This right here became the central hypothesis of the whole investigation. And it's an incredible idea when you stop and think about it. Could just 5 minuscule alterations in a single protein completely change how a virus behaves? It was time to gather the evidence and prove it.
So here's the crucial thing in science. A hypothesis just isn't enough. These scientists needed proof. So they designed a series of experiments, with each one serving as a clue to build their case and confirm that these 5 mutations were in fact the culprits. The first clue was a simple visual confirmation. They just repeated that initial experiment, but under super controlled conditions, mixing both viruses with turkey red blood cells. And the result was undeniable. The mutant virus consistently cut itself free much, much faster. This confirmed it had a clear performance advantage over the original. This wasn't a fluke. It was a feature of the new virus. For the second clue, they ran a performance test directly on the neuraminidase protein itself. You can think of it like putting a car engine on a test bench to measure its horsepower. They measured exactly how quickly that Na protein could process its target molecule. They wanted a precise number for its efficiency. And the real takeaway from these graphs is coming up next.
This chart boils that whole complex test down to one key number, the Michaelis constant, or Km. Just think of Km like a golf score for enzymes. Lower is better. A lower Km means the enzyme has a much stronger grip or affinity for its target. And as you can plainly see, the mutant virus has a significantly lower Km. Its molecular scissors aren't just faster, they're also way better at grabbing onto what they need to cut. Now, the final piece of evidence was maybe the most compelling of all. Using some seriously powerful computer software, the researchers created these detailed 3D models of both the original and the mutant NA proteins. And this let them move beyond just numbers on a chart and actually see the physical consequences of those 5 tiny mutations. And here's what that looks like in 3D. On the left, that's the original protein, and on the right is our mutant. Those lines you see show the molecular bonds that form when the protein grabs its target. Just look at the mutant protein on the right. See how it forms more of those bonds? This is the visual proof of what the performance test told us. It has a much, much better grip. We can even zoom right in on the effect of just one of the 5 mutations. A single change, this one called T149I, completely altered the surface of the protein. Just look at the difference in the shape and the charge in that highlighted area. This one small swap created a more effective surface, essentially fine-tuning the tool for better performance. It's incredible.
So, with all the clues on the table, the visual proof, the performance data, and those amazing 3D models, it was finally time to put it all together and officially crack the case.
The conclusion was crystal clear. Those 5 unique mutations, they weren't just random changes. They were a set of coordinated upgrades. Together, they literally reshaped the virus's escape tool, improving its surface, enhancing its grip, and ultimately boosting its efficiency. This allowed the virus to cut itself free from infected cells faster, giving it a serious potential advantage in spreading from cell to cell.
And hey, don't just take my word for it. Here it is in the researchers' own words from their 2024 paper. They concluded that this specific, novel combination of mutations was directly responsible for the physical changes they saw, the higher binding they measured, and the faster virus escape they witnessed in the lab. Case closed. Okay, so that's a cool scientific detective story. But why does the tale of one super efficient H9N2 virus matter to the rest of us? Well, this single case is actually a perfect snapshot of a much larger, ongoing global process. First, this is literally viral evolution in action. We just watched through a perfect real-world example of how viruses fine-tune themselves to get better. Second, it just hammers home that small changes can have a huge impact. I mean, 5 amino acids is an infinitesimally small change, yet it created a more efficient virus. And finally, this is exactly why the never-ending mission of global viral surveillance is so incredibly critical. By tracking these tiny changes as they pop up, scientists can start to anticipate how viruses might be adapting, and that helps inform things like vaccine development and lets us prepare for future outbreaks.
And that really brings us to our final thought. This study gives us a detailed blueprint of how a virus can level up its own machinery. It's a powerful reminder that in the microscopic world, this evolutionary race is always on. And it leaves us with a pretty critical question. If these 5 tiny coordinated changes can make one flu virus this much more efficient, what are the next million changes happening in viruses all over the world right now building towards?